This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 101, recorded October 1st, 2010. Hi, everybody. It's Vincent Racaniello, and this is TWiV. And we're back for the next 200, and we have a great crew today, starting with Dickson Despommier, who is not across the desk from me. Hello, Dickson.
Hello, Vince.
Dixon's in Fort Lee, New Jersey.
He is.
This is a historic occasion. It's the first time Dickson has ever used Skype.
Well, that's not true, but it's the first time I've ever used it on my computer. So
All right.
That's not true.
Welcome back.
Thank you. Sorry I
Next,
missed it.
next on the list from Western Massachusetts is Alan Dove.
Hello, hello.
Clear as usual.
Yes, and I guess the echo we're hearing on Dick is because he's shouting across the Hudson River.
That's right. Exactly. And it's a very cloudy and stormy day.
It
But
is.
where the next guest is from, or the next host is from, it is not cloudy and stormy, I'm willing to bet, from north central Florida, Rich Condit.
Hi, everybody. Great to be here. Yes, it's few puffy white clouds, 82 degrees.
Yeah, right.
We're
Nice.
finally out of the bad stuff. It's beautiful here. It's
It all
just
came
beautiful.
up here.
Yeah. Well, he means bad, meaning 100 degrees plus.
Right,
Oh, well,
exactly.
some storm just crept up the East Coast and nailed the East Coast really bad.
And in my office, our special guest today to help us talk about adenovirus is pulling him out of retirement, Professor Hamish Young.
Hello, everybody. Glad
Well-
to be here again.
Welcome back. Hamish, of course, used to be a professor in this department when it was called microbiology. Did you know now it was microbiology and immunology?
Yes, I knew that. Yes.
So Hamish has retired, and it's not a thing that many scientists do, right?
I guess not. I mean,
just
people
keep
look at me
working.
very quizzically and as though I'm some kind of mutant. But I'm quite enjoying my retirement. I miss not running gels and looking at sequences. And
How about plaques?
I miss plaques. Yes, I've got a story about plaques later on.
Oh, very good. Well, you spend the summers in Nova Scotia,
Yes,
right?
yes.
So, and I understand you like bird watching.
Yes, I'm a big bird watcher and botanist and amateur everything.
And when you come back to New York, do you still watch birds?
I watch birds in Central Park, and I'm a newly fledged, I guess that's the right word, member of the Linnean Society of New
Wow.
York. So I'm returning to my old love, which was natural history, from which I sort of wandered into virology a long
That's right.
time
You were
ago.
a zoology major.
I was a
right?
zoology major,
I
yes.
remember.
Yes, but I did an elective in virology in a forestry department.
Wow.
So
It's an interesting
you may
one.
ask why. Well, the forestry department was involved in insect control, and one of the things they were interested in was using viruses as biological insect control. And so tibula iridescent virus, et cetera, and that wasn't used as control, but the other similar viruses were used as potential insect control. And they had good virologists. So
And this was in
Oxford,
Oxford.
the head of Department of Forestry in those days. Maybe they still do. And that's where I saw my first pictures of icosahedral viruses, including
Electron
adenovirus.
micrographs
Yep, the Richard Valentine, the classic iconic picture
of adenovirus?
of adenovirus from
with
Richard
the
Valentine.
spikes coming
With the
out,
spikes
right?
coming out, which is, I think, almost unique because subsequent EM pictures have really not shown the fibers sticking out
Oh,
from
wow.
the vertices. How he did that is not clear. Must have been the fixation techniques or contrast techniques or whatever they're called.
It is an iconic.
It's an iconic
photograph.
picture. Yep.
I'll have to find it and use it. for
I
this
use it in my
episode.
lectures. I can give it to you.
Uh-huh.
Yeah, do that. I think that would be a good image for this episode. Well, we're pulling you out of retirement to do a little work here.
Okay.
It's not bad.
I promise to do my best.
Oh, I think you'll supply the color commentary. Because we have two papers on adenovirus, and I thought it would be a good time to put them together and have you join us.
Okay.
And the first is actually two papers,
Right.
which is amazing.
Yes.
The three-dimensional solution of the structure.
Right.
of adenovirus. And we should tell listeners how hard a thing this is to do because it's a huge virus.
It's a huge virus, yes. I don't know how much bigger than anything else before. I mean, most of the other icosahedral viruses have been maybe no more than a third of the size, I would guess. I don't remember the exact dimensions of polyoma and SV40.
They're considerably smaller.
Yeah, they're like 50 nanometers.
Right, so this is a...
900?
900
angstroms.
900
nanometers.
Yes, 90 nanometers.
90 nanometers.
Yeah.
Just how big is a pox, Rich?
Like 350 of the long way and 270 the short way, something like that.
The angstroms, right?
Yeah, but poxvirus, they don't count. They're just kind of out there.
The mimes are bigger.
Huge.
The mimes are out there too.
Right,
yeah.
right.
So this is a very large virus.
Right.
And I read in one of these papers, there's 1 million amino acids in the particle.
Yes.
That's amazing.
Yeah. Yeah, I think this is an incredible tour de force on the part of these two groups, and that they should come out simultaneously, I think, is also extraordinary.
Yeah,
right, because they're using two completely different techniques.
Exactly.
Yeah.
Exactly. I mean, the cryo-EM stuff has been going on for quite a while. Roger Burnett was one of the pioneers of that, and had actually built I think a 6 angstrom resolution. If I'm not. It might have been Phoebe Stewart. My names may not be getting the right attribution here, but the 6 angstrom one gave us a very good idea of what the overall structure was, but it didn't show the amino acid contacts, of course, because you need to get down to 3.5.
I think what they also did was to solve individual structures of individual proteins, right?
Again, Roger Burnett did the crystal structure of hexon back in the late 80s, maybe.
Right.
And then the fiber was done and penton base was done.
And those could be fitted into the...
They could be fitted into the
cryo-EM.
cryo-EM. It turns out from one of the papers here that the penton base clearly wasn't quite right, at least probably not quite right, because it wouldn't fit the fiber into it.
Right.
So there are very interesting structural characteristics of this virus, which I think are worth emphasizing for our listeners.
I would like to, before we do though, could we briefly explain the difference between X-ray crystallography and cryo-EM? The two techniques, each paper used a different technique. Right. Who can do X-ray crystallography? Is there anyone here that feels strongly? If not, I can read it from my textbook.
Yeah, that would be a very good idea.
So in X-ray crystallography, you make a crystal of virus particles, which in itself is a work of art, a little black magic. A crystal is made of particles arranged in a 3-dimensional lattice. Then you take these crystals and you bombard them with X-rays.
Right.
When you do that, each atom in the particle scatters the radiation, and the interactions of the scattered rays with one another form a diffraction pattern which you record. It used to be on film, and now you do it directly with a scanner. Each spot on this diffraction pattern contains the information about the position and the identity of all atoms in the crystal. That's something that's always been amazing to me. And the locations and intensities of the spots are stored, and then you have to figure out how they relate to one another, which requires a lot of other steps that we don't need to get into. But
a lot of math.
A lot of computing.
Fourier transform.
Fourier transform, which to me is just magical.
Yes, I don't know.
There was a woman here, you might have known her. Her name was Barbara Lowe.
Oh, yes, Barbara Lowe. Yes, of course.
She was an X-ray crystallographer. She was a good friend of Rosalind Franklin.
Right.
So she used to talk about this all the time at lunch, and no one understood her.
Right.
So the resolution of X-ray crystallography could be less than 2 angstroms.
Yes.
Right.
Yes.
1.5 or less, because the X-ray beam has high resolving power. And proteins...
the key thing here, of course, is to get the crystal, right, with the right
That's it. You
the
have
right
to get the
coordinates
crystal.
and the right
That's the hard part.
Right. With the right
X-ray
asymmetry.
crystallographers in graduate school would spend 6 years trying to get their crystal, and then in 36 hours they were done.
Right.
Yeah, getting the crystal is hard. You have to get the protein in the right conditions.
Right. And I think the key issue here with this particular paper is that they modified the fiber.
That's right.
So that you didn't have these things waving around in the breeze. One of the problems is if you have variability in the structure
Right.
that reduces the resolving power,
Right.
or makes interpretation more difficult. I don't know the exact
made-
terminology. So they popped in a very short fiber,
Right.
which sort of recessed into the particle a bit, which is an interesting thing which we might come to later.
And they did that by making a recombinant virus,
Yeah,
right?
they put AD35 fiber onto a backbone of, I think, AD5.
Right. Now, the issue of the resolution of these techniques is important because a protein alpha helix is a nanometer in diameter.
Right.
So you have to, that's 10 angstroms.
Right.
So if you're not less than 10 angstroms, you can't see the backbone.
Right.
And you want to be even less so you can see
Individual
the
side
individual
chains.
side chains.
Right.
And that's why the 1 to 5 nanometer resolution, angstrom resolution is what you want.
Right.
Now, cryo-EM is a very different technique where you take virus particles, but you don't crystallize them. You freeze them in water.
Right.
And you don't stain them. You just take pictures of them frozen under an electron microscope.
Right.
You take hundreds and thousands of pictures.
In this case, thousands.
The
right.
idea being that each photo is in a slightly different orientation, and then you use a computer to assemble them all.
Stack them up.
It's like a CAT scan, basically, right?
Right.
Except of viruses.
So if I understand it correctly, the resolution that you get is a function of this reiterative process.
Yes, I think the more you do, the better you get.
And they did something like 30,000 for this.
Yeah.
31,000 individual particle images in 1,300 films. It's amazing.
Yeah.
Now, this fellow who's the lead scientist, the PI on one of these papers, Hong Zhu from UCLA, I've just heard he is phenomenal with cryo-EM. I remember we were reviewing a grant once, and someone said, if anyone can do this, he can.
Yeah. Yeah.
So he's apparently very good. So
I'm really astonished that you can get this kind of resolution.
It is. I think,
It's
I mean,
amazing.
Harrison in his review says that the actual resolution in the cryo-EM is better in ability to trace the amino acid backbone than the crystal.
Yeah, I was actually amazed looking at these two papers, and I saw, there's an X-ray and a cryo-EM structure. Well, we know which one's going to be more detailed.
Right.
And then
It's the opposite.
Turns out not to be the case.
Yeah.
Yes, Harrison says the cryo-EM density map of the appears to be substantially clearer and more interpretable than the X-ray map, which is amazing.
It's extraordinary.
So it's 3.6 angstroms for the cryo-EM, and they can see all the proteins, the entire capsid.
Right.
So both papers have basically the same structure, but very different approaches. And I think it's great that they were published together. Right?
I think it is, and I think they're mutually reinforcing. I mean, they come to essentially, I think, the same conclusions. Slight details between the two which are different, but this is a major advance in the adenovirus field.
Hamish, these viruses, maybe we can start talking about the structure by talking about these viruses are composed of pentons and hexons. What are they?
Well, the hexons are the proteins that make up the faces of the icosahedron, and there's, I think, 720 of them in the structure. So there's a lot of them, and they're arranged in trimers, and then they're arranged in faces. And the pentons are the proteins that exist at the 12 vertices of the icosahedron. So they're a different protein. So it's not quasi-equivalence in the classical sense. There's a different protein at each vertex. And there are 5 of those at each vertex because there's a 5-fold axis of symmetry around the vertex. And sticking into that is the fiber, which is a trimer. So there's another issue here. So you've got all the faces made up of hexon, the penton bases which sit at the vertices, and then you've got the fiber which sticks out from the pentameric structure of the penton base. So you've got some interesting problems here. You've got a 3-fold axis of symmetry with the fiber going into a 5-fold penton base. So you've clearly got contacts which have to be dealt with. I mean, that you can't put 3 into 5 and have equivalency at each point. So there's something interesting going on there in the assembly of the virus.
In fact, I think Harrison said we still don't understand.
No, we don't understand that.
How the 3 goes into the 5.
Right. And that's always been a problem. I mean, how do you get 3 into 5? And how do you have, the principle back with Casper and Klug back in 1962 was that you made things as equivalent as possible so that you could self-assemble a viral particle called
Right.
quasi-equivalence. And this is clearly not a quasi-equivalent kind of situation where you've got 3 proteins having to intermesh with 5 proteins. So I think that problem is going to take a little more detailed resolution.
These hexons that surround
The pentons.
penton,
Right.
that strikes me as an interesting structural problem, right?
It is, yes, because they're making different contacts to the hexon-hexon contact. So again, you've got a quasi-equivalency going on.
Right.
And
So the structure of those trimers must be different than the structure of the trimers that are actually in the face. Is that correct?
That's what I assume. I had a little difficulty, not being a structural protein chemist. I had a little difficulty wading through all the various beta jelly rolls and alpha helical sheets, beta sheets and so on. So I wasn't quite certain how they resolved all that. I must confess my homework didn't extend to hours and hours and hours looking at this paper. So I didn't fully understand but there must be differences, I assume.
Must be.
Yeah, I think that is, and they talk about that. They talk about the contacts that are made between the penton base and the hexon versus the hexon versus hexon. So
did you guys look at these animations?
Oh yeah.
No, I did not. No, I
they're
didn't.
really cool.
Are they? Yeah,
Yeah,
the videos of the
They have a number of animations of both intact virions. So, for example, this is one of the intact virion. They rotate it.
Yeah.
You can see the hexons and the pentons.
Yeah.
The blue are some of these
accessory
what they
proteins,
call
the
minor
glue proteins. Yes.
There are how many of those?
Well, there's protein 3A, there's protein 8 and protein 9, which figure prominently in these discussions. There's protein 6, which is on the inside. Then there are 2 core proteins, protein 5 and 7, and mu, which are very basic and bind to the DNA. And there's some evidence, which they allude to, that there's a protein called 4A2, which is present in several copies per virion.
And I think the resolution of some of these accessory proteins is really the
I found that
particularly
interesting because I actually worked on protein 8 and protein 9, and the second paper, the cryo-EM paper, which goes into some detail about these proteins and previous. What I like to see in that paper was there's lots of references to previous work, which was very well documented in the Zhang. Is it Zhang?
Probably Zhang
paper. They actually refer to, for example, Colby and Zhao. The Colby and Schenck paper, which showed very clearly that protein 9 was important for the structural integrity of the virus. And then, of course, Frank Graham showed that you could stuff more in if you didn't have protein 9, because I guess the particle can get a little bigger. Well, back in the 80s... So
you can make viruses without protein 9?
Yes. One of the first host range mutants made in adenovirus, which was missing E1A and E1B, also took out protein 9. It's a virus called DL313, isolated by Jones and Schenck back in the late 70s. And he discovered that this was very, very labile. You freeze and thaw it and it sort of falls apart. I mean, it doesn't like maltreatment. So 9 was clearly but you can dispense with it. That is an interesting phenomenon. You can actually get rid of it and it still makes a particle.
Although they probably wouldn't survive in nature.
In nature, no, they probably wouldn't. Protein 8, it turns out that Jones and Schenck also made a mutant in protein 8. And we discovered that one was also extremely thermolabile. In fact, we published a paper in 1985 documenting that with Fred Volker, you may remember.
I do.
And clearly protein, this was an insertion mutation. And when I have a bit more time, I think I'll get in touch with Zhao and ask him where this would map and whether we can make some sort of sense out of what happened.
Oh, you got to come back to work.
Well, yeah, but I don't know where the viruses are.
Someone asked me recently where all your viruses are, by the way.
Well, they're in Saul's freezer, but he keeps on threatening to throw them away. So someone
came by and was looking for something. I don't remember who it was.
Oh, okay.
I have a question, gentlemen.
Yeah.
Yes.
As this virus is assembling inside a cell, obviously to go to another cell, these proteins are all being made in sequence or all together, or how do they self-assemble in order to go to the next level?
That's a terribly difficult question to answer, Dick.
Yes,
They are, I mean, all the late proteins are made more or less contemporaneously. Some of the non-structural ones are made perhaps a little tad earlier. But the nature of the assembly is still controversial. There are two opposing schools of thought. One is that you make an empty capsid into which you stuff the DNA with its associated core proteins, and the other is that you make the core and then you assemble the proteins around it. These structural papers may start to point us in a direction. One in the Zhao paper points out that there's some evidence that there's I've forgotten the name of the protein. It's a what's the word I'm looking for? It's a protein that shoves stuff into the particle. It's like a motor protein, a pump, a portal.
Packaging?
Portal
is the word.
Portal,
they
yes,
use.
portal. That's right, a portal protein, which will take the DNA and its associated core proteins and stuff it into the. And there are bacteriophages that do this, so this is not
and herpes too as well.
Yes, and herpes. Yeah. So it's not an unreasonable.
So, if with cloning technologies and protein sequences and expression vectors and all kinds of wonderful stuff at our disposal,
right?
Couldn't we just synthesize all the necessary proteins in the right proportions and mix it all together and watch what happens?
That would be the holy grail.
Has anyone tried that? Because I know.
Well, Dick. People have tried similar things in the past by isolating the proteins biochemically and trying to get self-assembly in vitro. And it's never really worked. You can get sort of subparticles and you can get this and that, but you never get the full particle.
So which viruses can you do that for?
Polio.
Keep going.
Probably with adeno, you need lots of cell proteins to assist.
Yeah, you might need chaperones. And stuff like that.
Yeah, you might well do. And you probably need these non-structural proteins like 100K, which is a non-structural late protein, and maybe 4A2.
So it's incredible to think about the evolution of these viruses then. I mean, look at all the accessory proteins and necessary help from the cell it needs in order to get to the next cell. How does all that stuff evolve?
Dick, it's a parasite.
No kidding. But I mean, this is incredibly complicated, isn't it?
But you could imagine sort of incremental steps getting more and more complex as you go along, starting from a simple self-assembling entity, which gradually gets bigger. And when you get bigger, you need to do other things, like having accessory proteins. So I think, how do you get the eyeball? It's the old story of Darwin, right? I mean, it's.
I'm not arguing against that. I'm just saying.
No, you're expressing wonder.
I am expressing wonder.
Yes, it's the Darwin. What is it? There is. What is the final statement in Origin of Species that Steve Gould used to use?
God has
there's a grandeur in this view.
There is a grandeur in this view of life, and I think that's what you're.
Totally.
Yeah.
Absolutely. Totally.
But other viruses have been assembled.
Oh yes, I mean, lambda, for example,
and other phages too. Not many animal viruses. So I think polio is the only one where you can translate the genome in vitro and get assembly of infectious particles.
And you don't get many, as I recall. It's a pretty inefficient.
It's orders of magnitude less than a cell.
Yeah,
it's not too bad.
Yeah,
right. And the enveloped viruses, of course, would pose a whole different set of problems.
Yeah, sure, exactly. Now, Hamish, one thing I got out of this was that these accessory proteins act like the N and C termini of smaller viruses.
Yes.
So the picornas, the SV40s, they are made of pretty similar proteins, but the constraints of assembly are such that they have to have slightly different interactions.
Right.
And that's mediated by the N and C termini, which can be flexible and assume different positions. So what Steve Harrison says is that these minor proteins, they're separate proteins, but they have the same function.
Yes,
right?
and but the interesting thing, so you might think the evolution would be that they were N or C terminal parts of the main structural proteins, but of course they're not. Protein 9 is miles away in genetic terms from hexon, penton, base, and fiber. Protein 8 is embedded in one of the late transcription families, but it is not next to hexon. I should remember this. I think it's hexon 6. No, it isn't. It's 6 hexon. Maybe it is next to.
Kathy is going to be clucking as she listens.
Yeah, right. Well, Hamish is losing his memory, but it's not the C terminus of hexon. So
here,
I
we got the map for you.
Thank you very much. That's helpful. So there's P6, 2, and 23K, which is not a structural protein. So P8 is actually in this L4 group, which is separated a long way from the C terminus of hexon.
Yeah.
So together, gentlemen, how many proteins does the virus induce versus how many it needs to assemble itself?
I think there are 11 or 12 structural proteins, and there are 3 or 4 non-structural late proteins.
And how many host proteins does it need?
Who knows?
Right.
Yeah.
Don't know.
Yeah.
So how would you go about exploring that? Knockouts?
siRNA knockdowns?
Yeah, well, I suppose you do the pull down experiments to see which cellular proteins are associating with which viral proteins. I don't know if that's been done with late proteins very much. It's been done with early proteins a lot, but and then you find out which ones they are, and then you sort of knock them down and you see if they have an effect.
Or you could do the shotgun approach and just do a library of CERNAs and knock down all the genes in succession. And see. Of course, that won't get you to combinations of them, but it would get you a library of potential.
Yeah,
does adenovirus actually induce protein synthesis in the host for host proteins?
There are changes in gene expression which happen after viral infection. Most of what people have looked at has been the early stage of infection, and that's true for both looking at protein-protein interactions and also for events that happen shortly after infection. So there are changes in host cell gene expression that occur upon binding of the virus to the cell surface, upon entry into the cell, and then after viral gene expression starts, there are changes to cellular gene expression. So it's an area that's been quite widely looked at early times. At late times, there's not a lot going on except for tons and tons of viral proteins being made.
Yeah,
so it's not an area. This is interesting. The history of adenovirus is that after a flurry of activity in the late 60s, early 70s, looking at late proteins, everybody focused on the early proteins because they contained the transforming transforming genes. So there was a huge industry of adenovirologists working away at their benches, looking at what E1A did and E1B did, and
including the former chairman.
Yes, right. So this was the growth industry. Late proteins lagged, and to some extent they still do. There's not a lot of people working on late proteins. So I think your point is well taken, Dick. I don't know the answer, and maybe a grant will be written to look at that issue.
One of the things that both papers and the review by Steve Harrison mention is that this information may be useful because adenovirus is being used as a vector for various therapies, and this could be used to redesign them or modify them.
Yes.
Antigenically. What do you think that's something that might happen? Or
well, there have been attempts to modify it antigenically because the variable loops of hexon, which are where the type specificity happens, those have been modified. Actually, going back a long way now, people have modified the variable loop once it became apparent that there was such a thing. To change the specificity and also to retarget the virus. You can imagine ways in which you could change targeting by targeting the fiber or by targeting.
So the fiber attaches to the receptor.
Yes,
right?
the knob is the thing that attaches to the receptor. So people have changed that with varying degrees of success. So I'm less. I mean, even though I was a sort of vectorologist latterly, I'm less enthused about that than about the more fundamental issue, which is that it may give some insights into assembly and also entry. Those I think those are areas that I think are really fascinating.
I found the justification a bit empty.
Yeah, I agree.
10, 15 years ago, they would not have had that. They would have said, this is a new structure and it illuminates icosahedral assembly.
Yeah.
But I felt they had, almost as if the journal made them include some practical information.
I don't know about Zhao, but Nemero is involved in, and his group, I think, are involved in vectorology. I think there was a note at the end of the paper which said something about a patent.
Scripps
right?
has a patent.
Patent on something. So, Scripps is full of sort of, I imagine there are spin-offs and so on.
Oops.
So, so I suspect there are lots of vectorology types that I find this interesting. And it is. I mean, there's no question that you can play around with the virus in fun ways, but the really fundamental thing, it seems to me, is that having the structure, and you know this from polio, once you had the structure, a lot of questions could be asked about fundamental virological questions, and that seems to me...
you can ask them very intelligently by tweaking particular places on the structure and get specific results.
Exactly. I mean, Alan, you know that.
I mean, one of these papers, I think it was Zhao who said, we now know how these proteins interact. We can change can change the interacting amino acids to improve or make worse the interaction, make bigger capsids or something.
Right. So I think this is a fundamental advance for adenoviruses.
Ultimately, most structural papers, I mean, as difficult as these things are to get out the door, when they're out, what you get is a foundation for future research.
Exactly.
It's a fundamental finding. It is a true basic research finding.
Exactly.
The problem is these days you can't really write your grants that way.
I guess so.
I don't know. I think you still should.
Yeah.
People are I think scared of it.
You probably need to include some little tagline in there.
Sure.
About
health.
we can tie this to gene therapy.
Yeah. Oh, by the way, we can do that. But the fundamental stuff is really important.
Yeah, that's where ultimately all of the clinical discoveries are going to come from anyway.
There are dozens of grants that can be written based on these structures, but I tell you, they will not do that well because you have to have this innovation as part of the proposal now. It's not enough to say this interface of two proteins is important for this. It just doesn't go. With money being very tight, they want something in addition. So, although I agree that this is wonderful and you can learn a lot, I think it would still be difficult to support. Not by me, but just by the whole process, the way it works. Rich knows what I'm talking about.
Yeah.
Hamish, could you, just for the novice virologists in the crowd, myself included, of course, I might be the only one actually, could you put in context this particular adeno 12 virus with all the other adenoviruses, and would it be an advantage to know a similar set of data for all of the other adenos? Could you then develop a family history?
I think this is adenovirus type 5, if I'm not mistaken. Certainly the Nemero one is. I don't know which one.
So Zhao is which adeno 5.
Adeno 5, right. So
they're both 5, yeah.
Yeah. So the other adenovirus, they come in sort of groups, and this particular group C, is the one that's most intensively studied. So 2 and 5 are the ones that were the sort of genetic babies back in the 60s and 70s and have continued to be the major source of information. There are transforming ones like AD12,
right.
And then others which cause interesting clinical problems. So the ones that cause ocular disease, and then others that cause gastrointestinal disease. So there's a spectrum of diseases caused by the viruses. The problem is that some of the others are not very easy to grow, and so you run into the same problem that you've got to crystallize these things, and they may not crystallize well. I mean, Nemero had to do this thing of putting a specific fiber. I assume that this was a deliberate choice to do this, or maybe they'd made it and it crystallized. I don't know which, but so you'd have to invest a lot of time and effort to reproduce these papers with another serotype. And I'm not sure that the life cycle, the biological life cycle, or the cellular life cycle is all that different. The interesting question with adenovirus, it seems to me, among the various serotypes, is how they cause different diseases. And I'm not certain that the structure is going to help you in that. The fiber and the contacts that are made between the virus and the first cells they infect, that can be done without doing a complete structure of the virus. So I would think that you could focus on fiber, you could focus on the variable loop of the hexon, and then you could focus on E3, which produces all these proteins that modulate the immune response, which Bill Wold has done for many, many years.
Also, you can take the approach that you would with any model system, which is, okay, we've got a structure of AD5. We'd like to know the structure of AD whatever.
Right.
So let's make the simplifying assumption that it's probably pretty similar to AD5, and let's spot check it with site-specific mutations.
Yes.
That would cause known changes in the AD5 structure and see if they cause known biological changes, the same types of biological changes.
Right.
So you can kind of infer those sorts of similarities.
Yep. I think that would be the way to go, rather than to try to reproduce these tour de force for other serotypes. I think that would be very difficult to do.
I was just thinking out loud to myself here, of course, in isolation here in Fort Lee, New Jersey, that 10 years ago when we had the Genome Project in full swing, and you start out, wow, we've just cloned the genes for E. coli and we've got the whole sequence now. And you build on that. And then, of course, everybody else has those genes too, or some variation of them. And now today they're thinking of, how many people are they thinking of actually doing the entire genome for? 10,000 people?
100,000.
Well, the difference is that the technique is so much easier.
Understood. Well,
I think Dick, that the issue here is still that to make a crystal of any entity is, as Alan said, you spend 6 years making your crystal.
Sure.
And 36 hours analyzing the results.
But now you don't have to make a crystal. You can do it by cryo-EM.
That's the point.
Yeah, but still, 30,000
images is
30,000 images
child's
is
play.
Not trivial, and it's not something that...
Neither was the base pair analysis trivial until they improved the machinery.
Yeah, I understand.
So maybe they'll come up with a machine that can actually, or machine, I say technology, obviously, to read all these things off very, very quickly.
My guess is that it wouldn't get past a funding agency.
Well, even more fundamental than that, I mean, the proteomics was supposed to have been the next revolution after genomics, and the reason it's taking so much longer to happen is that protein structures are just inherently orders of magnitude more difficult to work with than nucleic acid structures. And so, we'd like to be able to have structures of all of these things, but the technology is not there, and I don't anticipate it being at that point for decades.
Right. I mean, just how many picornaviruses have been done, Vince?
Over a dozen, easily.
A dozen, okay.
Yeah.
But that's over a period of...
Many years.
Since
1985.
20, so 20 years, 25 years. So,
they're much smaller than
and they're much smaller, and they've been crystallized for decades. So I think this is not I don't think that, Dick, this is going to be a growth industry.
I withdraw my question.
No. Actually,
it was an excellent question.
But what was thought about years ago was that at some point when we had a database of structures, we would then be able to look at a protein sequence and say, this is how it folds.
Right.
We are not there yet at all. I don't know how long it will be, but maybe that is a reality one day. Yeah,
that actually I would consider a little bit more likely, but it's another extremely difficult problem. It's apparently a mathematically very, very complex problem to calculate a protein folding from first principles, even if you have all of this vast amount of data to work from.
But I think your point,
chaperones figure into that as well, because not all of the information for folding is in the primary structure.
Exactly.
Hamish, is there anything else here that we should cover before we leave this beautiful piece of work?
Well, I think the only thing I want to say is congratulations squared to these people for doing such a beautiful job. I mean, I think both papers are landmarks. These are landmark papers. I think both.
It's also beautiful to look at. You know,
Yep.
I got my issue of Science and I opened it up and I read these two things.
Yep.
I didn't understand half of what you guys were talking about, but I appreciated the beauty of what they had done.
What's amazing,
Alan, the supplementary material on these is open access. Is that correct?
I believe it is. Yes, it usually
Because
is.
if it is, we can put the animations on the
sure.
on the website, right?
Right.
Yep.
Yes.
Yeah, we'll check that
So
out.
people can tweak them. The first two, the full capsid and one of the faces, are both really fun to play with.
The face, I don't know if you've seen this, Hamish. This is one face.
Yeah.
And they rotate it so that you can see the minor proteins, how they hold together the hexons. It's just beautiful.
I tried to access that from home and I
Couldn't get
failed.
Hamish and I are in the same technology.
Yeah, we're sort of Neanderthals when it comes to this.
That's right.
Yeah, it's beautiful.
Yep.
And there are not a lot of authors on each paper.
Yeah, I
know?
noticed that. Yeah.
Not many at all. Okay, that will do it for the structure of adenovirus. Let's move to another adeno story.
Which also relates to structure.
Yes, it certainly does. Now, we have talked on TWiV before about the possible involvement of adenovirus in obesity. This paper is from Blood. Is that the name of the
Pediatrics.
Yep.
Adenovirus 36 and obesity in children and adolescents. So there's last year, I think, a paper came out on this. So this causes a lot of activity in the press. Everyone picks up this paper. So what they did in this study was to identify 124 children. This is in California, in the LA area, and they're about median age 13 years old, and some of them are obese. They were studied, determined that they are obese, and others were not. And then they took their blood and looked for neutralizing antibodies against adenovirus.
Right.
And this serotype had been implicated previously.
Right.
They did a neutralization assay,
Right.
and what they find is that many more of the obese kids have antibodies to the virus than the non-obese kids.
Right.
That's basically it.
Yeah, and I think it's a pretty robust statistical difference. I mean, I don't think there's a problem there.
About half the kids, 46% were not obese and 54% were obese, which unfortunately means that they probably took a pretty representative sample of American kids.
Unfortunately.
Yeah, they say here that, 17% of children and adolescents in the U.S. are obese.
Right.
So this is, the numbers here, so 15 of 19 obese children had AD36 antibodies. And
no, they did it the other way. Let's see. AD36 was 19 of the kids total were positive for AD36.
Right.
And the majority of the ones who were AD36 positive were obese. 15 out of the 19 who were AD36 positive were obese.
Yep.
And the AD36 correlated with, if you look at the subset of obese kids with AD36, they were the most obese.
Yeah.
They had the biggest waists and greatest, all that.
So just to play devil's advocate, obese children are more susceptible to this virus.
That is precisely what I concluded when I saw this paper.
Yeah.
Or any virus.
Yes.
Correct.
In fact, in the case of adenovirus, I mean, obesity causes a host of respiratory problems.
And immune problems.
And it is entirely plausible, and I would even argue likely, that these kids are going to be more susceptible to a variety of infections.
Right. The only argument against that is a sort of peripheral one, and that is that people have infected various experimental animals with AD36 and shown a change in their body mass index. Mm-hmm. So that's partly where this is coming from. So I agree absolutely that correlation is not causation. But there is a background to this in experimental science showing that AD36 infections, I think in chickens and maybe rhesus monkeys, or they mentioned that.
Maybe.
I don't remember if it was mice, but there's certainly chickens and I think rhesus monkeys showing that there's an increase in...
primates, non-human primates, rodents, and chickens.
Yep.
Is it an increase in the production of growth hormone, do you think?
I don't know that the mechanism is known in those experiments.
It's also possible that you're looking at two completely separate phenomena.
Oh, absolutely.
And you can imagine that an infected laboratory animal is not going to move around as much.
Right.
So it says here that in the rodent model, AD36 decreases hypothalamic monoamine levels and is associated with a reduction in corticosterone secretion, which leads to impaired fatty acid metabolism.
Right.
So there is somewhat of a mechanism there.
Right.
So then this is an after the fact experiment. So what would you actually do to design the perfect experiment to show the correlation is 1-to-1?
Well, the correlation we already know from this finding that
you have to do this at birth, right?
We know already that it's not 1-to-1.
No, but you'd have to start a serological survey of kids from age 0 and follow their every move until they started to develop obesity to see whether or not this was actually correlated.
Well, isn't there a big cohort of children being followed? But
that's not a difficult experiment.
Is that a longitudinal study?
That is a longitudinal study.
That's what they say they need to do here.
Yeah.
Because this is a cross-sectional study.
Right.
So you'd have to follow them. And
a cohort of children
the sera
find out.
Exists for that.
Sorry?
The sera already exist for that in other studies that have already been done.
That's what I would think. What I would think.
Framingham study up in Massachusetts.
But that was for adults, wasn't it? I mean, it's not children.
Well, they did everybody, I thought.
Did they? I mean, there is a cohort of children who went through, I think, in Seattle, where they followed the infectious diseases that these children got. It's a classic study back in the 50s and 60s, I think. 40s and 50s.
Obese children.
No. All
children.
To find out, what diseases they got when. When did they get their first rhinovirus infection? When did they get their first adenovirus infection? If I'm not mistaken, there was something along those lines in Seattle.
But shouldn't such studies really do a survey of all infections in these children, not just AD36? Because we need to address the question of whether they're just generally more susceptible, right?
Yeah, I mean, I think this is a study based on a preconception, right?
Sure.
Since there's some experimental evidence that AD36 causes obesity, let's put it in rodents, therefore let's go and have a look at humans to see if the same thing might be true. So it's presupposing a mechanism, I guess.
Right, and in fact, the press release that went along with this completely stripped out any discussion of the causality versus causation, or correlation,
right?
And it's all focused on it looks like AD36 might cause obesity because of course that's what's going to get the headlines.
Exactly.
That's how you get the grant.
Yeah, the press release kind of feeds into the hype.
I think actually a study section, having sat on study sections where these kinds of things come through, the likelihood of it getting funded is close to zero.
Depends on the study section.
I mean, our study section looked at those correlation things and said, bleh.
Yeah, but this will go to a clinical medicine study section. Very, very different criteria.
They'll get a grant from Twinkies.
No, I don't.
If it don't cause obesity, then that's great, right?
Well, Dickson, it may not cause all obesity. It may be responsible for a fraction of it, and there's dietary considerations as well.
You know what study would actually be very relevant for this is if you went back and looked at historical serum samples, just any population of historical serum samples that you've got, preferably of U.S. Or some other fat country, and go back 30 or 40 years if you can.
Before obesity,
before this had reached epidemic proportions in the U.S., when it was unusual for people to be obese,
exactly,
and see what the prevalence of AD36 was 30 years ago.
Now, let me just pop in a bit of adenoviral sort of nonsense here. AD36 means that it was one of the later serotypes to be identified, because the numbering system is based on when they were first identified.
And there are 50 some odd.
There are 55 now, different serotypes. So 36 is a fairly late member of that group. Now, that suggests to me that it wasn't terribly prevalent back in the early days of adenovirology.
Yeah, you had to look around for it.
Yeah, and so that's a factoid. But I think it's worth remembering that it certainly wasn't the one that Wally Rowe or Hillerman found back in the
if that's true, Alan's data will show that it wasn't there, and then you can.
Well, it would be a little more plausible, perhaps.
Sure.
Right.
This is interesting. I was at a meeting yesterday where they were discussing a collection of 9,000 human sera taken in 1950. Someone went and looked for hepatitis C, and they found it in a fraction of the sera. Mm-hmm. And, of course, Hep C not discovered till the 70s, right? 80s, early 80s. So you can do these kinds of studies. And, of course, in that study, they had to go back and find the people and say, hey, you might have Hep C. But that could be it. But I also would like to see a longitudinal study looking not just for AD36, but even other adenoviruses as a control.
Right. Yeah.
And at the same time, some good cell biology where you see if there's a real mechanism for increasing.
There must be something like that. With all the attention that's been focused on obesity in particular, there must be something like that going on now.
Well, the last time we talked about it, I looked, and there wasn't anything very convincing. There's nothing in Journal of Virology or Virology, for example.
The best no, not that would necessarily. I don't mean that would necessarily have generated the virology data you're looking for, but some kind of longitudinal study of kids and diet and obesity and that whole thing, where there would be a cohort and the samples would be taken, and there'd already be an archive of samples, but maybe nobody's looked at the viruses in those samples.
Yeah, they're probably doing it now.
There's a whole other set of studies. Sorry to interrupt here, but I'm reminded that there's a whole bunch of studies now going on in the pediatric field to look at the gut flora and how that differs between obese and non-obese people, and they find huge differences. So, it's possible that the uptake of nutrients is regulated by some subset of microbes that makes you more efficient, perhaps, at storing molecules you needed to run away from the rhinoceros that was chasing you, but we're no longer being chased by rhinoceros, so therefore...
or your uptake of nutrients regulates your gut microbiome.
Indeed.
And those studies which were done also in mice that can show that transferring the microbiome associated with obesity confers obesity to the recipient mice.
That's right. Cook's postulate, so to speak.
So we don't do that in people, obviously, but you can do it in an animal model. Then you can look at the epidemiology. And that's being done. People are looking at obese microbiomes.
One of the points they made in the paper is that the obesity epidemic is very fast. I mean, it's happening very quickly. And some comment about it, diet has changed, but has it changed that much and that radically to cause that epidemic? I've forgotten the exact phraseology they use. I think it's in the early part.
Yeah.
The diet has changed much slower than the epidemic has occurred.
Yes, I think that's the point they're making.
That's absolutely. It's not correlated at all.
Only by dietary excess coupled with decreased activity.
That's right.
Yeah. So I think there is something to this obesity epidemic which is not currently being identified.
I think it's multifactorial.
Well, of course. Right.
There's diet and television.
I think supersizing is a big part of it too, right?
Sure. I think it's
the plate size gets bigger,
Right. The portion size has grown dramatically, and that has happened in direct correlation and introduction of higher amounts of processed food. And you can point to many causes, and I suspect the truth is going to be that all of them have contributed a little bit.
The internet, computer games,
high fructose corn syrup.
Yeah, I think that I've heard that's another component as well.
Interestingly, in Europe, I didn't find anything with high fructose corn syrup.
No, it's not used there because they don't have to dispose of the huge glut of corn that we have.
That is.
I thought we use it for ethanol for fuel.
Oh, we use it for anything we can cram it into.
That's true.
Because we're paying our farmers to grow too much of it, but that's not a virology topic.
No, I'm sorry. Well, this is interesting because we like to blame microbes for things, and with AIDS, HIV-1 is the cause, but it's not the cause of everything like this. And there are many diseases that are probably multifactorial.
Right.
So that's an interesting one, and we've been talking about other diseases which probably fall into this category as well.
Yeah.
All right, we have reached the hour mark. Shall we save the last story for next time?
Sure.
Okay, let's do a few emails here. The first one is from Russ, who sent us an image from a website called 3d4medical.com. He said, I think this image is great, and this is an app for the iPad from which he got it. So let me show you this image, Hamish. It's a very... Dick, you should...
Yeah. Oh, it's great. Yeah.
I'm going to send it to you on the Skype link, Dick.
Oh, good man.
Because this is a beautiful image that you'll like to see.
Yes.
And it's isn't that nice?
Yeah.
We'll wait till Dick sees it. You see, the link is popped up there, Dickson. Click
Just
on
click on it. Okay, I'll do that. Oh, look.
What is that,
Really
Dickson?
like that.
Very, very cool.
It's a green-eyed monster. It's a female mosquito.
Yeah.
And I'm convinced that this comes out of Tony. I'll think of his last name. I'm sorry, but he actually got green fluorescent protein to isolate into the eyes of mosquitoes genetically.
Uh-huh.
So Tony James, his name is Tony James. In fact, he's a National Academy of Science member because he was able to do this, and he employed Drosophila promoters in order to get the genes to work.
Cool. How do you know this is a female?
Well, it doesn't have whiskers on its.
Dick, come on.
She's
It's
feeding.
feeding.
That's also
we may
true,
be
of course.
virologists, but we do know what a mosquito does.
Oh, but do you see these antennae?
Yeah.
They're not hairy like the males.
Oh, really?
That's another
Oh, okay.
difference.
That's how we
All right.
call that the whiskers on the males. So you're absolutely right. Of course, she's feeding.
Yeah.
That's right.
All right, thanks.
That's a Far Side cartoon.
That's a cool. Anyway, the website too, which we'll put a link to, has some other interesting images as
Yeah,
well.
it looks like a great website.
We have next one is from Julian, who writes, "My name is Julian.
Ruiz
He's a-
Science.
Ruiz Science, DVM, MSc in virology, and PhD candidate from the Universidad Nacional of Colombia." And he says South America, because we wouldn't know.
Well, Colombia, you see, with an O.
Colombia.
Yes, not Colombia, right? After some discussion with virologists, including Dr. Anne-Lise Hanny, business secretary of the International Congress for the Taxonomy of Viruses, we wrote a discussion paper about the living nature of viruses, trying to travel from the virus history to a new concept and findings about viruses. The paper was published in the last issue of Acta Virologica. It's called Viruses, Virophages, and Their Living Nature. And I would like to share it with the biggest team of virologists on the web. Thank you.
Well, we should send it to them.
Does he mean by size or by volume?
Anyway, we'll put the paper on the site. It's a very nice paper.
It is a nice paper.
And he says, keep online podcasting. You have faithful listeners on this tropical virology area. I hope to meet you in ASV in Bozeman. Well, Julian, I hope I met you there.
Right, because it's happened.
This
That
is how
happened.
far back we go on our letters. Yes. Sorry.
And then he says, as a pick of the week, I would like to share the best virology book that I have ever read, The Invisible Enemy, A Natural History of Viruses, from Dorothy Crawford, professor of virology from the University of Edinburgh, which is, I've learned to say it properly.
Correct, Vince. Good.
I said Edinburgh for the longest time, and a guy actually sent me an email from Scotland. He
Good.
said, please, you're driving me crazy.
Yeah.
Edinburgh. That's a pretty good book, I understand. I haven't read it yet, Julian, but we might pick it someday.
Yeah.
And
So I have to interject.
Yes,
one
do.
of the papers that he references in his article is this Nature Reviews paper, which you subsequently told me you'd blogged on, Vincent. But of course, I don't read your blogs, so
that's okay.
I hadn't seen it. But it's this Perspectives in Nature Reviews called 10 Reasons to Exclude Viruses.
Yes, from the Tree of Life. Yes.
So then,
And-
I love it when you bring up a PubMed abstract and there are 6 comments.
Yeah.
And
Yes, that's what I really loved was the comments. This created a
Let's see.
huge flap.
see what
Okay.
those comments are.
Oh, they're fantastic. There's one from Lynn Enquist. I should actually credit this properly because it's I think its first author is somebody in his lab, Ludmir and Enquist. And there's another one from Dieter Rualt, who the discoverer of Mimi virus, and another one from Eugene Koonin.
Oh,
who, of
great.
course,
Yeah.
So really created this huge flap.
Fuss.
And it's and they're all very different, and it's a terrific discussion of this ongoing discussion we've had about whether viruses are alive or not. I think it's Coonan at some point, who uses a new word that I really like. He refers to them as undead.
That's right.
Undead. That's good.
Zombie life.
They're zombies. That's right.
Yeah.
But,
That's amazing.
And Dieter Rualt goes off and says that he really likes, for a definition of life, he favors Shakespeare. A tale told by an idiot full of sound and fury
Signifying nothing.
So this is a wonderful series of comments.
This is good.
That's great.
This is excellent.
I read the paper last year when it came out, and I didn't know that there were all these comments. So thank you, Julian, for pointing that out to us. We'll put links to all this stuff.
And his paper, Julian's paper, has a lot of history of virology in it as well. That's very readable. It's very nice.
Yeah, he did a good job. I was looking at that, the history of that and how memes fit in and why we have to rethink this because of memes. Very good. All right, Matt writes, I love your show and have been listening since November 2009. I know that the topic of viral DNA being integrated into the human genome has come up before. My question is, could viral DNA that is in the genome from hundreds to thousands of years ago be expressed and produce a virion? Could it be possible for a virus to acquire genes from another virus that is in our genome? Thanks for the podcast and all the great work you put into it. He says y'all. Y'all put into it because Matt is from South Carolina. So, mouse endogenous retroviruses are still active. They make virions. Mm-hmm. The human ones happen not to be, but other species make, and yes, it could. And I don't see why it wouldn't be able to reassort or recombine with another virus in the genome.
Yeah, it makes sense to me. All that stuff is possible.
It is.
And of course it could, I think what his question is getting at is, could it reawaken? And I think that's possible.
Well, there's a human endogenous retrovirus that has been reawakened. We talked about it.
Yeah.
The HERV-K, which Paul B. Nash reawakened by fixing the sequence, and he made a retrovirus out of it. So
a truly undead virus.
Yeah, in fact, go back and listen to TWiV with Welkin Johnson. We talked about this a bit on that. This was probably an email from before then, Matt, and you've probably already got an answer to your question from that. But such as is that we are so slow at answering emails. The next one is from Alex, who's from the University of Leeds, also in the UK, right? Yeah. Hello.
Which is pronounced Leeds.
Leeds. Yes, Leeds.
Thank you.
You got it right, Vince.
Thank you.
Yeah.
I am an undergraduate microbiologist at the University of Leeds, and I wanted to thank you for your marvelous podcast, which I discovered last night. We recently broke up for our summer holidays, or vacation, as I've heard you say, across the pond, and I am finding both TWiV and TWIP very interesting, so thanks for that. You know, when I first read this, I saw broke up, and I said, oh, my God, he broke up with his girlfriend over this podcast.
That's an Anglicism. It means that they went on their holidays.
Broke up.
Yeah.
So they don't use that for relationships.
No. Well, they may do, but broke up means you stop going to classes and go to holidays.
It's like food having gone off rather than disappear.
And you don't call them vacations over the holidays.
Holidays, yeah.
Now, he says, I thought I would let you know that the article in New Scientist mentioned in episode 1, titled Welcome to the Virusphere, was what initially tweaked my interest in microbiology and led me to study it in higher education. It seems only appropriate that it is mentioned again as I finished my first year of study. Dickson, you remember episode 1?
I do.
There you go.
I do remember it.
I do. You're doing a great job with the podcast. Please keep it up. Okay, and one more from Kathy, who is a high school teacher and TWiV enthusiast. I was so excited when I started listening to TWiV 87 because I have been a part of Dr. Hatful's phage hunting program. I am a high school teacher in Illinois and was accepted to the first teacher outreach program and spent a week in the summer of 2006 at the University of Pittsburgh learning the protocols for isolating phages. The goal was for teachers to get their students involved in authentic research all over the country. That first year, I had 8 students in 3 classes who found their own phage. One of them came in after school to finish isolating it and getting it ready to send it to Pitt. She named the phage. It was sequenced, and we were able to annotate the genome. It was then submitted to GenBank under our names.
Outstanding.
This was very exciting to me and to my students. I've gone back 3 summers and have trained 3 years' worth of students to phage hunt. While there, we got to listen to lectures on current research in phages in bioinformatics. We also worked closely with Steve Crisson on bioinformatics, so they are reaching more than just undergrads and local high school students. The grant has been renewed, so I will go back as long as they'll have me.
That's great. That's wonderful.
Yeah, nice success story.
Yeah.
Thanks for telling us that. Yeah, we did an episode with Graham Hatfull some
yeah,
time ago.
Yeah.
That was fun.
Yeah.
All right, let's do a couple of picks of the week. Let's start with Rich. What do you have for us?
So we've done some Richard Feynman in the past, and I've got another one. I've been scouting around for this for a while, and finally found a good site that from which you can view a 1981 BBC Horizon interview video where they interviewed Richard Feynman. Just to reiterate, Feynman was a Nobel Prize-winning physicist and quite a character who was very articulate about a variety of different things. And he wrote books on how he was interested in science, how he got interested, his opinions about lots of different stuff. He was, how do they say it? Marched to the beat of his own drummer.
It was an icon.
He talks, among other things, just to give you an example.
I believe he actually played his own drums.
That's true, too, yeah. He talks in this video, just as an example, about how he actually resigned from the National Academy when he found out that it was just a bunch of people who sat around trying to figure out who was cool enough to be in the National Academy Club.
Excellent.
So it's the video is 50 minutes long, but in particular, if you're interested in the kind of personalities that make up Nobel Prize-winning scientists and why people do science and how their heads work, it's absolutely wonderful. It's really good. Actually, the website looks good, too. This cosmolearning.com has all sorts of stuff. It's an open access learning site. It's very nice.
It's cool. The pleasure of finding things out.
Yeah.
I just searched for David Baltimore, and they don't have anything. Oh, that's great. I like He's got a great Brooklyn accent, too.
He does.
Feynman?
Yeah.
Oh, yeah.
Yeah.
Alan, what do you have for us?
I have from the National Geographic website. They had a photo contest, and they have now published the best environmental photos of 2010.
Oh, cool.
And the thing that drew my attention was the first photo, the overall winner, which was just absolutely jaw-dropping. I looked at it, and at first I didn't understand what it was, and then when I figured out what it was,
manta rays, I guess.
Look at them
absolutely amazing.
Oh
They're Monk's devil
rays. Gosh.
And it's an aerial view of a school of these rays swimming in the Sea of Cortez off Baja.
Yeah.
And it just goes, the depth of it just goes down. There's just layers and layers and layers of rays down into the ocean, and just beautifully composed. Cool. Anyway, there are some other good shots in there.
They're beautiful. I like this third one. I cannot believe the underwater world finalist, birthplace. That whole picture is underwater.
Yeah,
it's amazing.
Do you scuba dive?
Yes.
Okay. Have you ever dived in fresh water?
Yeah, springs around here, but they're nothing like this.
Yeah, if you have fresh still water, like a quarry, accumulates algae, and you get these flooded. Especially if you've got flooded trees or something, it's very, very.
I guess the closest thing I've seen to this is diving in a kelp forest in California. That's pretty amazing.
But usually the visibility is crummy. I mean, to get a clear shot like this is just.
This is just an amazing picture.
Dixon, I just sent you the link so you know what we're talking about here.
I've just gone online for their Nat Geo, so you're right.
Nice.
Beautiful.
We've got some other things out there, though, that are quite amazing. I love National Geographic. I think it's fabulous.
Speaking of environmental,
yes.
This is not my pick.
No.
But did you know that very recently a microbial mat was discovered off the coast of Chile that is the size of Greece?
Whoa!
Whoa!
It's like the fungus that was as big as Michigan, right? This wasn't
That's true. They're fungi that are hundreds of miles in size.
Right.
But an underwater microbial mat has been found in fairly shallow waters off the coast of Chile, and it's the size of Greece, or 132,000 square kilometers.
About the size of Alabama.
It's composed of various bacteria living in symbiosis. And you can read about that at Small Things Considered. So I thought I would mention that. But that is not my pick. My pick is a summary of the first International XMRV workshop, which took place a few weeks ago in Maryland. And this is a summary put together by the folks over at the CFIDS Association. And they did a great job of summarizing everything that went on. I don't know if any of you have heard about this meeting.
Yes.
Anyway, it's a great summary of all the sessions, multi-page.
What does it stand for, XMRV?
Oh, XMRV, xenotropic murine leukemia virus-like virus. It's been implicated in prostate cancer and chronic fatigue syndrome.
Okay.
They have all the talks summarized, John Coffin, Myra McClure, Robert Silverman, Kate Bishop. It is a 11-page document, very well done. So if you want to see what went on and where this area is going, check that out.
I like the fact that it was written by people from the, what's it called?
It's the CFIDS Association of America.
Chronic Fatigue and Immune Dysfunction Syndrome Association of America.
Right. So this
I think it's really nice that they wrote this.
Yes.
Okay. So they have credibility, I would assume, with people with CFS, and they've written a really nice summary of this.
Yeah. It covers the state of the science as we've discussed on TWiV.
Yeah, so that's cfids.org, and you can see there's a link on the front page, the first International XMRV workshop, 43 reports from researchers all over the world, and there's a link to the summary. So check that out. It was written by Kimberly McCleary, Stephen Kleinman, and Suzanne Vernon. So that's my pick of the week.
And Kimberly McCleary is actually the president and CEO of CFIDS.
That's correct.
So, great.
Before we wrap it up here, I want to send you over to virology.ws. There is a survey there to tell us how much TWiV you want. I recently put that up, and we have about 150 replies so far. I want to know if you want it once a week or every other week. So far, we have 134 responses, 59% every week, 41% every other week. Tell us what you want. I'm just curious. And there are lots of good comments down there. One fellow said, I want it every day.
I want it all, and I want it now.
And others have other comments as well. One interesting comment, if you try to do it in less time, it will become a news program and less of a teaching tool.
Yes.
PROGRAMME. It's probably from across the pond.
Yes, it would be.
Yeah, I want it to be a teaching tool, so we don't want to short things up, but just tell us how often you think it's worth having. We're happy to do it once a week, but I'm curious what you would like. And of course, if you want to listen to TWiV, you can do it on iTunes, at the Zune Marketplace, with Stitcher Radio on your smartphone, or at twiv.tv, where we have show notes and letters and all sorts of goodies. Check it out. If you're new to TWiV, leave a review in iTunes. It helps us stay on the front page of the medicine podcast directory. Besides twiv.tv, you can find us at microbeworld.org. Send us your questions and comments, twiv@twiv.tv. Alan, good to have you back.
Always good to be here.
You can find Alan at alandove.com. And Rich Condit, thank you.
Great to be here. You're quite welcome. I enjoy this.
University of Florida, Gainesville, home of the Fighting Gators. Hey, is football season begun? I guess so, right?
Big game this week. This is the big one.
You've already had games though, right?
Yeah, we're
even
coming.
I know that.
I don't know.
This is Alabama coming up, though. Yes.
This is Alabama coming up. They're number one. This is the real test.
You knew
this
that,
is
Hamish?
like game.
No, I
game
didn't know
5.
that, but
All
I
right.
knew they'd had football games.
I don't know.
Even
Well,
in Paris, they were talking about football.
But Rich, you go to these things. You have a season ticket, right?
I do. I share season tickets with Dick Warner.
So tomorrow you were
This is an away game, so I won't be going to this tomorrow.
Which Moyer
Watch it
used
on
to
TV.
be here, right?
Yeah.
Yeah, that's right.
Send Dick Moyer my very best.
And
I shall.
Hamish, a former professor who now lives in New York City and likes to watch birds. Thanks for joining us.
It was my pleasure.
We will have you back more frequently.
Well,
all right?
I'm happy to come
Keep
whenever
you
you
sharp.
ask me.
We like to have your input, and we'll keep you thinking.
Okay.
I don't think bird watching makes you think, right?
Oh, it makes you think a lot.
Yeah?
Oh, yeah.
Dixon,
sure.
do you bird watch?
While I trout fish, yes. When I'm not catching fish, I'm watching birds.
I see.
So he's watching birds a lot of the time.
I'm mostly watching birds. That's true. I have to admit that's true. I love watching birds.
From Fort Lee, New Jersey, Dickson Despommier.
Yes, sir.
Thanks for joining us, Dickson.
It's a pleasure, as usual.
Now that you have Skype working, maybe you can join us more frequently from Fort Lee.
Perhaps. Or I will show up in person again, of course.
Hey, Dixon's book is being published in a few weeks. The Vertical Farm.
Great.
Ah.
Check it out. It's on Amazon for pre-order. What's the full title, Dickson?
The Vertical Farm, Feeding Us and the Rest of the World in the 21st Century.
Yeah.
Endorsed by none other than Sting.
This is correct.
Excellent.
He has a quote on the front cover.
Yeah.
Yeah. All right. Thanks, everyone. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.